And good evening, and welcome to Chapter 27 from Tutor and Derrickson. You are listening to the slideshow of Bio 212 for Norwalk Community College. And what we're considering today are what are the body fluid compartments, and what does that actually mean to the body's ability to regulate electrolytes, and I forgot the C there. It's not working the way I want it to. And pH, electrolytes, and acid-base acid. I'm just going to go AB. I don't want to run out of space. Fantastic. Okay, let's see if I can go in. Whoops, where'd you go? Woohoo. Electrolytes and AB. Okay, in lean adults, okay, that would be, not me, that is for sure. Body fluids constitute 55% of the female and 60% of the male total body mass. And what I need you to consider is where you actually find these fluids. It's important to understand you should distinguish between intracellular fluid, okay, which is inside of all of the cells, hundreds of trillions of cells if you include the bacteria. You have fluid inside of those, and it makes up about 2/3 of the body fluid you actually find. Extracellular fluid, which is where we spend most of our time thinking about things today, is that space, the interstitial fluid between cells, as well as the plasma and blood and lymph, cerebral spinal fluid, synovial fluid. But you get the idea. How does it actually distribute itself? You can see here when you're looking at a cartoon of a female and a cartoon of a male, 45% of the materials are solids inside female versus 40% for males, and fluids make up that 55%. And what this means is, and you're thinking about thermogenesis and the ability to regulate temperature, you can see that as water is a thermal conductive tool, meaning the H2O inside the body, male temperature seems to be a little bit more stable and female temperature seems to be a little bit less stable. And the significance of that, of course, is the energetic needs to maintain temperature inside the organism. Okay? When we're thinking about here, that total body fluid, remember, most of it is intracellular. Please don't make this mistake during an exam, right? When you're thinking to yourself, what's the difference between intra and extra? Well, intracellular is inside the cell. Extracellular fluids, okay, are going to be outside here, but remember, if I drained all the blood out of your body, it only makes up 20% of all of the fluid inside of your body. And what we mean when we say fluid balance is, of course, there are two barriers that separate the interstitial fluid and the plasma fluid, right? And that's because you actually have plasma membranes of all of the cells, plasma membranes as well as blood vessels, which are, of course, plasma membranes as well, but they have a specific construction, and they also have smooth muscle as well as a basement membrane. So each of these create a slightly different gradient that would allow then for materials to pass inside the body. Because remember, anywhere there's a solute, the solvent will travel to dilute it, and that's actually what we're doing inside the body. Over the course of the semester, though, what you've seen is processes of filtration, reabsorption, diffusion, okay, and osmosis are the continual exchange, okay? The continual exchange of water and solutes among the compartments. If you think about how I always would draw a capillary bed, let's throw in some color here for a minute or two. How I would draw a capillary bed so that you could see materials moving in, bifurcating, increasing the surface area, okay? And then the consequence to that, of course, is that thinking back, whoa, what happened there? What we're doing is we're creating opposing gradients, right? Where you actually, in blue, you'd have an increasing concentration of carbon dioxide in this direction, and in red, you'd have a decreasing concentration of oxygen inside this direction that's available to exit from the hemoglobin inside of the red blood cells passing through here and to travel to the mitochondria inside the cells that will use it for inside the electron transport chain. Okay. So let's get rid of that. Now you see what I mean by continual. Okay, so where do the sources actually come from? Well, before I actually tell you where the water actually comes from, you need to remember that we've spent essentially the entire course, both AMP1 and AMP2, teaching you that you have 4 types of tissues. Two that are excitable and 2 that are non-excitable. And remember, the excitable ones are neuronal cells, meaning the actual neurons themselves, as well as the 3 different types of muscle tissue: cardiac, smooth, and skeletal, right? And remember, to make them excitable, we had to create a range of electrolytes on the outside and the inside of the membrane of that cell. But remember as well that all cells have some balance of electrolytes on the outside and the inside, and it's going to be primarily our kidneys that are going to be responsible for excreting excess water so you can actually have the correct concentration of materials inside the plasma of your blood as it's being delivered. Okay, so long-winded explanation, sorry about that, but body can obviously gain water by ingesting it. And remember, the product of the ETC here, so let's put that right there, TC gives you equals ATP plus H2O. H2O. Oops, it's supposed to be an O. O. Right? The carbon dioxide was the decarboxylations that we saw in the earlier steps of glycolysis and the Krebs cycle. And that's the metabolic water we're talking about. That's the whole point of writing that out for you, right? Okay. Now, you can lose water, 1,500 mils a day, being filtered and excreted from the kidneys. You can have evaporation from the skin, depending on what you're doing, where you are. Exhalation from the lungs. 300 mils seems like a low number for me, only because if you're inside, say, a air-conditioned building, essentially that's a refrigerator, and that actually is a desiccator. It takes the water right out of your body. So every time you inhale and exhale, it's taking incrementally more water out of your body. And then finally, to get the feces out of your body, it has to have some level of water inside of it. So that takes about 100 mils of water a day. Okay. So here's a table you have inside your textbook. And there's nothing really to be gained from this other than what I just explained. Notice the colors here. Blue is water gain, red is water loss. I don't imagine I'm ever going to give you a figure like this that says, well, what are the numbers here? The whole point, of course, is if you look at them, they should be roughly the same, right? Okay, maybe they don't exactly add up, but in reality, because some days you're going to be dehydrated and some days you're not going to be dehydrated, some days you're going to be really good and you'll drink 4 or 5 bottles of smart water, but you get the idea. What we want to get to is how do you actually regulate that? Okay, mainly by the volume of your intake as well as how much you actually lose. And when you're losing water, right, you have a decrease in the volume inside the body of water. I mean, at the end of the day, that's all it is. The consequence, though, that's what you need to understand, is an increase in osmolarity. And that increase in osmolarity is going to stimulate the thirst centers inside the hypothalamus. Okay, we've been here before. You know what's going to happen. Your mouth is going to get dry. You're going to stimulate the osmoreceptors inside the hypothalamus. And you're going to have a decreased blood pressure. And that's where our story picks up. Because hypothalamus, let's throw this inside here, is going to respond most basically by simply increasing ADH. Right? Over here, though, look, we get to release renin, and that means we get to work on our RAA pathway again. And for those of you who are not inside this class and have not taken AMP2 with me, if you don't understand the significance of the RAA pathway yet, just let me say that you need to know it inside and out, upside and down, and backwards. And the consequence of understanding how ADH and the RAA pathway work together, okay? Notice they're not working antagonistically here. They're working together so that you increase the total amount of water that stays inside of your body, okay? In this cartoon, though, they're giving you a new responsibility of angiotensin II. So today, okay, you're going to understand that angiotensin II stimulates the thirst centers inside the hypothalamus. So not only does angiotensin II work as a potent vasoconstrictor, where we considered it in chapters 18 and chapters 26, now I want you to understand that as you increase the amount of angiotensin II, you actually increase the thirst. Okay? Let's get rid of that cartoon. Let's go over here. So how do we regulate it? Well, obviously, elimination of excess body waste is going to be through micturition. Let's throw that big fancy word in there. Micturition. Micturition. Which is peeing. Okay? And to the extent of urinary salt is the main factor that determines blood volume. Thinking back to the Jack Daniels jalapeno popper, deep fried, salted food. Remember I asked, did people have like crusty eyes and all of that? Well, if you have so much excess salt inside the body, when you wake up in the morning, your body has to excrete it wherever it can. And if you're asleep, you're not peeing yourself. Or if you are, we don't want to know about it. Okay? And what I want you to understand is that you feel swollen when you wake up because the body needs to retain that much more water to dilute that salt. Okay? There are the main factors that determine the osmolarity is the amount of water loss, because if you lose more water, you actually increase the osmolarity. And there are 3 hormones, our 3 friends. We can't get rid of these guys all semester long. Okay, we have angiotensin II and aldosterone. And remember, these were made from the RAA pathway. Okay, and what's going to happen, of course, they're going to cause the reabsorption of sodium and therefore chloride when you're dehydrated. But, whoops, where'd that go? Whoops, where were I? But if you have too much water inside the bloodstream, it allows the secretion or excretion, would be the best way to put this, right? Excretion into the tubules so that you secrete it into the tubules, excrete it out of the body. So sodium and chloride will follow it to actually decrease blood volume. So let's take a quick look at this as we approach this is actually a really busy slide, and there's a couple places I want to make sure you're paying attention to. Obviously, over here is going to be if you have increased blood volume, or, okay, so let's go like this. You're going to, let's change color here. We're going to go like this. And the arrow moving downward is what happens to our blood volume if we have excess water and sodium chloride inside the body, right? So if you increase your intake of sodium chloride, you're going to increase the concentration of that sodium chloride, which means the osmosis into the interstitial fluid increases, and therefore you've increased the blood volume. What are you going to do? Well, because that blood volume has stretched the atria of the heart, we're going to increase the amount of atrial natriuretic peptide inside the plasma of the blood, and it's going to go target the kidneys. So you can allow for the removal, okay, of sodium and chloride. Notice it says here reduced reabsorption of sodium and chloride, which is going to be the loss of sodium and chloride. And then you're going to actually increase loss of water in urine by osmosis, is going to decrease the total blood volume. Now notice that didn't address what happened to the cells that lost all that water. It just addressed what happened when you actually had too much sodium chloride ingested inside your body. Likewise, on this side, if this is ANP versus ADH, over here you're going to see decreased release of renin in the juxtaglomerular cells. So in total, you're going to decrease the RAA pathway. And by that I mean activation of the RA pathway, which means you have less angiotensin II and therefore less aldosterone. So, what do we know? Let's get rid of the cartoon. My drawings here. What do we know about regulating normal water loss? ADH, as I told you guys inside class, there's some amount of it inside your circulatory system all the time. And the more the more dehydrated you become, the more ADH you actually have inside your bloodstream. So this would be a gradient, and this is you over here, hydrated. So let's go high. And this is going to have you over here, D, as in dehydrated. Okay? And this would be the concentration of antidiuretic hormone inside the bloodstream. And here's what you need to do. Notice you need to see that for us to increase the number of aquaporin 2 channels that allow for the recollection of water, we are targeting a specific cell population. Nudge, nudge, hint, hint, wink, wink. That specific cell population are principal cells. So ADH targets the principal cells, transcription translation occurs, we make more aquaporin 2, and then what's going to happen is it's going to increase the permeability, and therefore we retain water, AIN, right? H2O, but we let all the salts, Na, etc., out, and that's actually how you produce that concentrated urine from the, if you're thinking about how the juxtamedullary nephron functions. Okay, so let's get rid of the cartoon. Go like that. We go down here. The consequence, of course, is you're going to have movement of water. And what I want to make sure you're understanding is that just in general, if you think about this cartoon, should hopefully not bad flashbacks from AMP1, you have a normal red blood cell in an isotonic solution, you have a red blood cell in a hypertonic solution, and you have a red blood cell inside a hypotonic solution. Swollen, so let's just put S, crenated, and normal. And remember, our red blood cell in normal shape is biconcave disc with increased surface area, so we can allow for oxygen and hydrogen protons and carbon dioxide to move in and out of the surface area easily. Okay? So what are the electrolytes? I'm going to just get rid of the cartoon. I'm going to go here. Well, electrolytes inside the body, they are ions that are dissolved and dissociate as a result of the water inside of it. They can carry electrical current. They also serve as cofactors, meaning the increased efficiency of enzyme function occurs as a result of the presence of those cofactors. Okay, so notice I said presence of cofactors. Bold, underlined. Okay. It is primarily the major mechanism by which we move ions between membranes that will control the flow of water. I mean, for us to control water, we control the flow of ions. Okay? The engineering is such that you move the ions, therefore you control water. The consequences, we're going to maintain acid-base balance. Okay? And that acid-base balance, of course, we think about this cartoon for the umpteenth millionth time in this class. Right? I go like this. Okay, hopefully you see homeostasis when you see that. And remember, we have a range for, say, pH 7.45. Okay, there's a point there. And there's 7.35. The moment I put that up there, you know that what we're going to do is we're going to control the movement, say, here of hydrogen protons. Okay? Make sense? Okay, cool. I hope so. Let's see. Let's get rid of that. And we can do this. Now, I'm running up against my 20-minute timeline here. So all I really want to do is explain how concentrations are measured. And then we'll take a quick pause and we'll get on to the next piece of this lecture. We measure them in terms of what we call milliequivalents, MEQs per liter. And what this is essentially a millimolar concentration of the ion inside the solvent. And the solvent, of course, is water. And there's a chief difference between two extracellular fluid compartments, and that's plasma and interstitial fluid. The plasma of the blood, okay, has more colloid osmotic pressure because of the protein anions. Think about what hemoglobin does. I would say albumin does, or any one of the clotting factors inside the plasma of your blood. They allow for blood colloid osmotic pressure, and if I just draw this for you, here is a glomerular capsule. Yay, I shouldn't have picked this color. Well, I'll do it in red and I'll change. Glomerular capsule, proximal convoluted tubule, right? And inside here, remember, blood moves out as a result of hydrostatic pressure pushing things out. And it's going to hit the capsule, so you have capsular hydrostatic pressure. But because all the fluid moved out, all the proteins that were still inside this space cause the colloid osmotic pressure to pull materials back in. And that's a pretty good place to take a pause. Okay.